We're going to take a look at genome screening and sex determination. Embryonic screening is a form of genome screening. We can screen a person's genome to look for genes that may make the person more susceptible to genetic disorders. There are several advantages to embryonic screening. So one of these is financial savings. So you might get a reduction in disorders that need to be treated, and this would save a significant amount of money in the long term. During IVF, most embryos are surplus to requirements anyway, and in this case, screening would merely ensure that the implanted embryos are healthy. So during embryonic screening, we can screen embryos to see their genome and work out if they have any genetic markers which are associated with diseases. People can now have their genome screened to look for genes that cause the individual to be more susceptible to genetic disorders. However, there are several disadvantages to embryonic screening. One of those is discrimination. There is the potential that defining the desirability of an embryo based on their genetic makeup could promote discrimination. For example, people born without the use of screening could be considered second-class citizens. There are also some ethical problems. The embryos that are not used during IVF are often destroyed, and this can be considered unethical or against religious views. It's also very expensive. In the short term, screening is an expensive process, so this means it wouldn't be accessible to everyone anyway. And could we be using money for other things? If screening was mismanaged, this technology could become a way for parents to select the most desirable embryo as their child to be. So this has led to the term of designer babies by some people. So healthy human body cells contain 23 pairs of chromosomes, only one of which is responsible for determining sex or gender. This pair of chromosomes are called the sex chromosomes, and they can be either an X or a Y. Males are XY, so a sperm either contains one X or one Y chromosome. Females are XX, so an egg contains one X chromosome. So depending on whether the sperm that fertilizes an egg has an X or a Y will determine what sex the baby is. So if the sperm carries an X chromosome, then the baby will be a female because it will be XX. But if the sperm cell has a Y chromosome, then the baby will be a male because it will have an XY chromosome. And we can work out these using a Punnett square.